Dear students, welcome you all on the behalf of Rahul Anand tutorials. So as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand tutorials. So now let's begin with the very interesting topics which are often being asked in terms of your Neet, often the Aims, often the All India Pre-Medical Examination. So these are literally belonging from which of the chapters of your book? So these are belonging from the Biomolecules. So now let's understand what are actually these mean the biomolecules. So what are biomolecules? So biomolecules are actually the chemicals as well as what? As well as the molecules which are produced by the living organisms as well as being present in terms of where as well as the compounds or either the molecules or either the chemicals which are present in terms of what? In terms of the cells. So it will be coming under what? So it will be coming under the biomolecules. So now let's try to understand what are actually these with reference to the best example. So the example will be actually what? So it will be the protein as well as the here means the carbohydrate as well as what? As well as the lipids. Often you are having the vitamins etc. So these are nothing but coming under what? So these are coming under the case of the biomolecules. So often when we are trying to understand what are actually the carbohydrates. So what are actually the carbohydrates? Can you tell me? So carbohydrates are the optically active polyhydroxyaldehyde as well as what? The polyhydroxy ketone. Now, so it is often being further divided into the three broad categories. So what are actually these? So these are the monosaccharide. Then what? Then the polysaccharide. Then what? Then the oligosaccharide. So fine. When we are talking about the monosaccharide, it means what? It is the simple sugar or either the carbohydrate which cannot be broken down into any of the units or either simpler sugar. It will be coming under what? So it will be coming under the monosaccharide. Then we are having the here means the polysaccharide means what? The compound as well as the carbohydrate which could be broken down into what? 2 to 10 units of the simple sugar. It will be coming under what? So it will be coming under the here means the polysaccharide. In third we are having what? So in third we are having the oligosaccharide. So the carbohydrate which cannot be broken down. Sorry. So the carbohydrate which can be broken down as well as what? Or either the monosaccharide which could be broken down into what? Into the 10 to more units. It will be coming under what? So it will be coming under your polysaccharide case. So now here in these we will be understanding about the protein case in the. So now let's understand about what are actually the proteins. So what are proteins? Proteins are the polymer of amino acid. So now let's try to understand about these means what are actually the amino acid. So fine. Now, when we are talking about the amino acid, you have to understand about the two facts. So it is particularly composed as well as made up of the two things. Means what? The amino plus acid. So here, in this case, we will be having the two functional group. One of the acid as well as one of the base, etc. So here it is what actually? So it is the NH3. Means what? So NH3 will be what? Amines. And here the functional group will be of the amines. And after that you are having the carboxylic group. So it will be what? The COOH. So you could often find popularly here on the board here. Now, so when we are talking about these means the amino acid, it is often being further divided into the three broad categories. So these are actually what? So these are nothing else but the alpha amino acid, the beta amino acid, then what? Then the gamma amino acid. So based on the structure means what? Of the amino acid in terms of the position as well as the structure of what? The amine group, then the carboxylic group. It will be divided means what? Here mean the amines in terms of the alpha amino acid, the beta amino acid, often what? Often the gamma amino acid. So you could find here on the board here the alpha as well as the beta as well as the gamma amino acid. So now let's understand about this means the structure of protein. So it will be often further divided into the four broad categories. So what are actually these? So this will be the primary structure, directly proportional for what? Directly proportional for the 1 degree structure. Then what? Then the secondary structure, directly proportional for what? 2 degree structure. Then the tertiary structure means the 3 degree structure. Then in the fourth we are having the quaternary structure. Of course the 4 degree structure. So now let's understand about these in brief. So when we are talking about these means the primary structure of the protein. So what do you actually understand? So it represents what? So it represents actually the here mean the sequence of amino acid. Fine. Now after that you are having the secondary structure. So secondary structure represent what? Amino acid. Then in the second you are having the secondary degree structure. Directly proportional for what? Directly proportional for here mean representing the shape in which the long polypeptide chain can actually exist. So you know about the polypeptide chain very well. So what are actually these mean the polypeptide chain? These are literally being found where? So these are found in the DNA as well as often in the RNA. So in case of the RNA, mean the ribose nucleic acid, you will be having the single strand of the polypeptide chain. Often you are having the nitrogenous base in it, of course. So these are what? So these are the here, mean the adenine, guanine, then the cytosine, often the uracil. Fine. As compared to the DNA, we will be having what things? So we will be having the two polypeptide chain, often being attached with reference to the nitrogen base pair. So these are actually what? So these are the adenine, guanine, cytosine, as well as the thymine. So thymine being replaced by uracil, RNA with single strand. Fine. Now, so let's understand about the polypeptide chain. So these can actually exist in the two broad categories. So these are actually what? So these are the alpha helix structure. Then what? Then the beta plated CP structure. Now, so when we are talking about these means the alpha helix structure, directly optional for what? So you could understand in the DNA we are having these mean the alpha helix structure. Often these are particularly attached with reference to how much of the bond angle. So it will be attached with reference to the here mean the 24 angstrom. Of course, often it will be having the helical structure. Means what? The turnover in terms of the 3-degree structure. So 3-degree structure actually means what? So it means the here means the diagram which can stand with reference to the x-axis, often the y-axis as well as the z-axis. Fine. Now, so here means the DNA in terms of what? The alpha helix structure. So beta plated CT structure is often like that, but we will be understanding about that in our next lecture. So what are actually your mean the here tertiary protein mean the 3-degree structure of the protein. So it represents what? So it represents actually the overall coiling, then the bending. Fine. So overall coiling as well as the bending could be abstracted with reference to these in terms of what? In terms of the protein. So here you will be having the HN bonds as well as the CO bonds, literally in terms of our protein case. So we will be studying about that structure in terms of the brief. Now, so it is often being further divided, means what? Here, tertiary structure of the protein into the two broad categories. So these are actually what? So these are the fibrous protein as well as the globular protein. Now, so when we are talking about the fibrous protein, you have to understand about the certain facts. So what are actually these? So these are the water insoluble protein, often particularly present in terms of the long polypeptide chain. Of course, as well as what happens, it will be attached to one another by the help of the linkage, literally present into the point 1 to point 2 side. The best example with reference to these means what? Means your fibrous protein will be what actually? So it will be your hairs. Often what? Here mean the nails. So these are actually coming under what? So these are coming under the case of the keratin proteins, directly proportional for the fibrous protein. Now, in the second you are having what? In the second you are having the globular protein, directly proportional. By the name it is very clear that globular. Okay. So something will be soluble in terms of the water molecules. Fine. Now, best example with reference to that will be what? The pepsin, the renin. Now, so when we are talking about the pepsin as well as the renin, so you know about the renin role very well, playing important role in terms of the what? The coagulation of the milk. Often the renins playing important role in terms of the economizing as well as neutralizing, often breaking down and synthesizing of the fat molecules. It is now. So let's proceed with reference to these. Means what? Here, the fourth. In the fourth, mean the 4-degree structure of your protein. So in this case, what happens? So in this case, more than one polypeptide chain will exist. So fine, you know about the polypeptide chain, directly proportional, found in terms of what? Deoxyribose nucleic acid, often in the ribose nucleic acid. So now, so in terms of your quaternary structure, we will be having what? We will be having more than one polypeptide chains, mean actually two or either more. So you know about the polypeptide chain, directly proportional, found in terms of your deoxyribose nucleic acid, often the ribose nucleic acid. So, mean the two polypeptide chain or either more will be present in terms of the quaternary structure. So these are actually known as what? So these are known as the subunits. Find here. So now let's understand about these proteins in the brief. So these will be actually 20 in number. So these are actually what? So these are nothing else but the here mean the alanine. Then what? Then the arginine. Then what? Then the asparagine. Then the aspartic acid. Often you are having the another things in it, such as what? Such as the cytosine having. So after the cytosine, you will be having the here mean the glutamic acid. Then what? Then the gliadin. Then what? Then the glycine. After that you will be having here mean the histidine. After that what? After that the isoproline. Then the proline. Then after that you will be having the proline. Likewise the process repeats. So you could often find here on the board here. Here. After that, what things? After that you will be having the MPPS, then what? TQV. So these are actually the formula. Means the we will be having the methionine. Then what? Then the here mean the phenylene. Then the proline. Then what? Then the likewise the process repeats such as the MPPS. Mean the serine. Then what? Then here mean the MPPS TQ. Mean the threonine. Then the threonine. As well as what? As well as the here mean the thymine. Then in the last you will be having what? In the last you will be having the valine. So remember, so this is somewhat the condition with reference to your protein. So you could often find here on the board here. Till here position. Now, so the best example with reference, so you could understand that in term when we are having the word such as the RAM. So it is generally composed of what? The three words. So literally it could be written as well as demonstrated under what condition? Under actually the three different position names. So these are actually what? MAR, as well as what? RAM, as well as like the third one. Means what? Three way of writing these letters. Likewise, these protein arrangement are literally present where? In terms of our bodies. Fine. Now, so in this case, you are often having the protein. Best example in terms of our here means the sickle cell anemia. So when I am talking about these means what are actually the sickle cell anemia. Means what? So anemia is actually what? The bloodlessness. Fine. As well as the sickle cell means what? So the cell literally will resemble the case of the sickle cell. As well as primarily it was what? It was in the here. Mean the normal shape. So normal shape of the RBC or either what? Or either the cell will be what? So RBC are actually what? These are actually often the cells. Now, so these will be present in terms of the normal structure as well as resembling what kind of the here mean the structure. What structure? It will be present in the biconcave cell. Fine. Now, when we are talking about the defected cell, in terms of what? In terms of the sickle cell anemia, what will happen? It will change its shape and become what? It will change its shape as well as becoming what? So it will become concave in nature. So you remember concave in nature. Result means what will happen? The course as well as the management of the blood circulation mechanism will be defective as well as the here in the red blood cell which are becoming what? Which are becoming concave in nature could not here transported with reference to the different geographical regions either from the thoracic cavity towards abdomen or either from the abdomen towards the thoracic cavity or either the vice versa. Then what will happen? There will be the accumulation with reference to that area. Fine. Remember? So we are having the lymphatic system generally as well as often the blood capillary. So these will literally transported with reference to that mean the red blood cell will be transported as well as the region where it actually provide the nutrition will particularly swell down as well as the sequence with reference to our body will be disturbed. So this is somewhat the case of the genetic mutation means what? The sickle cell anemia. So we'll be understanding about it in our next lecture. So I thought we have got the concept what are actually the protein. Often what? Often mean the structure of the protein. Often you are having the amino acid as well as what are actually the polymers of what? The protein. Now, often you have understood about what are actually the demonstration regarding the alpha protein as well as the beta protein as well as the gamma protein. Often we have understood what? Often we have understood about the 20 different types of the protein here. Now, so you could often write these. So if you like the work with reference to the education, often the lecture. So you could often subscribe, like as well as comment below in the text. Thus I will be knowing that what concept actually you are hungry for or what haven't you understood or either got. So very thank you with reference to what? With reference to the here mean the Rahul Anand tutorial. So very thank you.